Yeah. So as the education coordinator, I mean, I meet with Tanya pretty much once a week, sometimes more than that, depending upon what's going on. And we're putting lots of effort into making these meetings and interactions sort of valuable to you guys, but we also view them as just really critical because you know, unfortunately with COVID, we haven't had a chance to do our boot camp and meet everybody in person. And you guys are, I guess, kind of becoming the Zoom graduate generation at some level. And you're kind of getting used to this, but you know, they're not as fun as meeting in person. We just hosted some in-person events at TAMU, both with Ariane Zees, who visited, and then we did a recruiting thing at the beginning of this week. And it's way more fun to be in person than it is on Zoom. But we will be meeting in person hopefully soon in June through a boot camp, which we're planning to hold in College Station. But I just wanted to sort of emphasize the importance of these meetings and really including them in your schedule and treating them kind of like going to a class, basically. Tanya works hard to sort of make the material relevant and sort of keep everybody working together. And one of the things I think too that's been great, I've worked with both Chris and Arani on their IDPs recently, and there's a lot of work that's gone into that, but I can already sort of see tangible benefits from going through that process. I think certainly Chris and Arani would agree with that, and I think it's helping us sort of be more focused and organized. I've never used IDPs myself in the past, and like I said, I can already see a massive benefit to using them. And Arani's co-chaired with Greg Sorden, allows the three of us then to have common ground and have something to both sort of plan forward for, but then also, you know, next year we'll be able to review and say, you know, did we meet the sort of expectations that we, you know, agreed on, both as mentors and mentees. So again, just sort of to reemphasize the importance of making sure this is on your schedule. The other thing too that's really important to us is that, and you know, Tanya, it's something that we can do maybe a little bit more regularly, or try to, as we sort of become more established, but to also get feedback from you guys. And if at any point there is things that you would like to see tweaked or modified, or even if you just want to say, hey, I really enjoyed what you did in that last training session, it's great to hear from you guys. And it's important to the leadership of BPRI to make sure that you guys see the benefits of sort of having these meetings. So at any point, if there's something that you would like to see tweaked or done differently, or have suggestions for improvements, you can always contact Tanya, but you can also always contact me or any member of the education committee.
Okay, thank you very much, Ben. So I will go ahead and kind of start our agenda. So
okay, I'm gonna go because I've got another meeting I need to go to. So have a good session. And again, thanks for taking the time to be here and to be here regularly in the future.
Thank you, Spence.
Bye.
Bye-bye. Okay, can you guys see my screen? Okay, so today, same as last time, we are going with the research presentation. But before I start, I need to give a quick overview. Especially today, we have 3 presenters. One will be an undergraduate student. So the level and how they work is different. So please be mind when you are commenting about their research or their work, because it is really very diverse background. He's from urban planning, like completely different background to our locus main graduate students. Okay, so another thing is I have already shared with you the memorandum of understanding. Thank you for who look into the documents and provide feedback, and we are going to implement it from the next month, from the 1st of March. So Spence will send the final version to your PI, and PI will ask you to sign it. Okay, so if anything that you're not happy with that, please let me know before 1st of March, then I can edit it to the documents. And the next thing is, I think some of you have received this email from the Dr. Gabani, our project leader, asking any comment for the BPRI strategic plan. You don't need to comment, but I wanted to emphasize that we are trying to get feedback from our trainees. Then we can, like, you guys are engaged in every step. That is what we need to achieve. So next is, it is a kind of, we are trying to update our website to look nice, actually, I think, because I know that millennials are very good in navigating each website and figuring out what is the best way and what is our bad in our website. So if you can scan this QR code, it will go to the Google Doc file. So when you have a time, please visit to the BPRI website. And if you see that, this is so ugly, this needs to be changed this year, and this need to add some information, or do you think that new outlook or new information that should be in our website, please let us know. Like, we are trying to change our website before boot camp. So we are now collecting all information. I highly recommend that trainees feedback to get it for this one, because you guys know how should make the website more interactive and kind of attractive the trainees. The thing I'm telling that to you is, from the next fall, we are recruiting undergraduates, 9 students. So it should be, for them to, it should be like kind of feel, wow, this is cool kind of thing. So your feedback is really needed for that sense. And the next one is Odelia and Vivian from Texas A&M. They are doing a very great job updating our social media. If you haven't following us in the social media, please follow us. This is our link at NSF-BPRI. So please follow us and like and thumbs up or save something like that. I don't know. So from the bridge, I learned that for Instagram, if you save that post, it considers has a high impact. So if you can save some posts here and there, it would be great to, at the end, to report to the NSF. And today we are starting with the presentation. We are going with Chris from TAMU, PhD student, and Doris, and I think Chase will join. If not, it's okay. Let's try to figure out. And how the presentation will go is, Chris will, or the student will explain for 5 minutes, and everybody have another 5 minutes for the Q&A session. And if you have like a specific event or need to share some information specifically, I think you can directly email to the speaker too afterwards. Like if you have very specific feedback or like to share some research article relevant to their research, you can be done like that way. And after this presentation, we have another presentation or short presentation about the ESA symposium. It will be delivered by the Arani. So now I will stop sharing my screen and allowing to Chris to start.
Okay, let me. I'm assuming you guys can see the presentation. Okay. So basically, overall, the first part of my experiment is to see where the intake targets of the various species of Schistocerca, where they're located. I put up some basic past examples of what has originally been done, of what an intake target geometric framework chart looks like. So basically, the idea is I'm going to be following what Simpson and Robbenheimer did back in the day, where they have, 2 macronutrients, and then they measure the consumption of a locus in solitary phase and gregarious phase and compare them. And you can compare across species as well. Basically, you just give them you do one that's a no choice, basically constraining them to one diet, and then another test of a choice to where they're given two options and they can feed freely. So you can have like a high carb and high protein, and they can have a little bit of both. They might have more of one, more of the other. And that's basically what this chart in the top right of these four show. Is sort of a theoretical approach of how they get to their intake target, with the orange being one diet and the blue being another. You can see that they eat a little bit of one, they eat some of the other. And so this has been done, like I said, in gregaria. And that's great. And we want to do it for the other species as well. But I'm also changing up the approach in which we do it, because while the information is insanely valuable that we have, there definitely are some improvements that should be made. So, okay. So basically, this is what traditionally the experiments have been like. So they'll take a solitary individual, put it in a cage, and then give it the food sources, and take a gregarious individual, put it in its own cage, and give it the food sources. And if it's a choice test, they're given two foods. If it's no choice, one. And they're allowed to feed freely. However, if you think about basically their phase as, let's say, a continuum, you can be like a spectrum, you can be fully solitarious, fully gregarious. But the problem is if you put a gregarious individual on its own for a long period of time, there are arguments to be made that it's not actually exhibiting realistic gregarious behavior when it comes to feeding, as well as other things. And so I want to improve upon that by doing what they've done with all the species, including redoing it with gregaria. But go to the next one. Yeah. But what I'm doing is I'm adding two more steps. So like I said, if you think of it as a spectrum, we have on the far left a fully solitarious individual, and then on the far right, the most realistic, replicable gregarious environment. So the third one, third chart here, is basically an example of, it's one large arena, and each locus is given their own section. So the dotted line is sort of a fencing. And the reason we have it like that is so they can interact, they can touch each other, see each other, smell each other, all that, but they don't have to compete for any food. They have their own food sources, but because they're interacting, that may impact where their intake target is. So it might actually be different than them being on their own, whether that be because they're moving more or because of how they interact. We don't know yet. And so to continue that even more gregarious, that represents more of a realistic environment, is they're interacting. So they're given the two food dishes like they are before, or one ratioed food, and they're allowed to freely feed. And so this creates a lot of interactions between them, not only physically, but competition. And basically we're going to see how that impacts everything, thinking that they're definitely moving a lot more than they would in the previous cages, being together. I am also, for the choice test of the free gregarious, the one on the far right, I'm going to have a camera mounted above it, so we can basically track each individual, not only to measure consumption, which is a big part of it, because it's like we don't know who eats what. So that will help us measure the consumption, but it will also tell us a lot about their behavior, which I think will be really interesting, and hopefully we'll get a lot of information out of that. This is basically the exact same. Before was like sort of a graphical version. This is what it actually looks like for each of the cages that I've built. And yeah, I mean, that's pretty much it. Just trying to, if you think of it as a continuum, trying to get each spot and to see how the intake target between protein and carbs changes for solitary to gregarious and all along that. So yeah, that's that's pretty much it.
Thank you, Chris. So now, anyone have question or feedback or would like to share something else?
So is the solo gregarious your control?
Kind of in a way. So I guess, yeah,
Okay.
it kind of, it is like a control. Yeah, I'll say yes.
And then are you gonna do something in reverse with the solitarius one, like put a solitarius in with the gregarious and see if their feeding behavior changes there?
No. So the reason I'm not going to do that mostly has to do with time, because each experiment takes 2 weeks full time, and with 4 sets per, it's just, I do want to do more, and I want to actually, these are all, I didn't say this before, these are all final instar nymphs. I actually do want to compare it to adults and maybe reproductive adults, but that would again be if time allows. But that would be really interesting to see how it would interact and would change with if you have different phases, but just time crunch, so.
And also, are you just going to be doing it with three of the locusts? I was just thinking if the sample size would be big enough for them to have that tactile and chemosensory and all of that factors that you're trying to incorporate in your experiment. And also, just to follow up for the second question, are you going to be redoing that for the solitary as well, or just changing up the dynamics for to measure the feeding behavior of the gregarious phase?
Yeah, good question. So I'm only doing it for the gregarious individuals, the different sort of setups. And when the solitarious individuals, they're wrapped so they can't see others. So it's to basically make sure it's fully solitary with no interactions. And then yes, it's going to be 3 per arena, with 3 in the segregated one and then 3 in the free roaming one. And that's to keep it, the reason it's like that is to keep it per area, like per square inch, the same. So it's basically standardizing space. And that's with the arenas we have. 3 is the best it works. And they do have, based on the prototypes I've done, do interact a lot, even in the fenced off versions. Whether it will change their actual intake, not quite sure yet. I'm hoping that due to their interactions, if it doesn't, that's valuable. But they're also moving a lot more and like climbing a lot more than they would in the standard arena, based on what I've seen. And so I'm hoping that will show something, but if not, then yeah. So there might not be enough to create an interaction, but I guess we'll definitely have to see. Yep.
Is that all? Does anyone have any questions?
Maybe I missed this, but what is your hypotheses? Like, is there going to be higher protein feeding in the gregarious? But like, I think this ties into what Ariane does in her work, but I don't know if I missed that with your talk.
No, I kind of like mentioned it because I'm not 100% sure, but my hypothesis definitely is that as it gets more to the realistic, fully gregarious phase, that there'll be a higher carb intake than, like, compared to the solitarius or even the solo gregarious, just based on their energy needs. I think that a combination of stress between being together in a cage and also them moving around a lot more, having more space, free roaming, they'll basically need more carbs than they would protein to be able to maintain everything that they're exerting. So that's my hypothesis, but it very well could be wrong.
Yeah, because I mean, if you would have seen, I think it's from Spence, but I'm not sure, where they have the geometric framework for both the solitary and the gregarious locust. And if you see, the solitary one is kind of more, it's not as convex as Locusta migratoria, but it is slightly more convex than the gregaria. So I mean, that still tells that even when they had gregaria separated, it was still eating differently as compared to the solitaria when they were on confined diets, because the gregaria one is rather more linear, like we see, it's like that, but the solitaria is more of like it's not as convex, like I said, but it is still moderately convex.
For sure. And that's why I think the no choice are like super important, maybe there'll be more, I don't know if they're going to be more nutrient maximizers or error minimizers as they go along the spectrum. I still imagine that sort of layout, like you're talking about, will stay the same between the differences between the two. That just based on thinking about it, that's what I would say. But again, don't know what it's going to be like when they're interacting. Hopefully trying to replicate more field-like environments as much as you can in the lab, and sort of getting that. But yeah, I'm hoping that there'll still be the difference between like nutrient maximizers and error minimizers.
I have general comment. Maybe it's not applicable for your kind of research. What I learned from my PhD when I'm doing that after analyzing my data, I based on the human data, but my data was different than what my hypothesis is. So my statistical method I planned was not able to do that because data is not valid for that. Then I have to go to the one statistician in the stat department, ask what is the test that applicable for that. Then she said, you have very limited choices because your experimental design cannot accommodate this technique. So she asked, when you're doing something again, when you set up your experiment, get some advice from statistician, whether if this cities get wrong, do I have second chance to use my data? That is what I learned from my PhD. So I will encourage you to, just in case things get wrong, what will be my second option? Think
Okay,
about
yeah,
that.
yeah, definitely.
My one article still I'm struggling to publish because of that, because human data definitely cannot be normal and it is really hard to get whatever statistic applicable for the journals. Okay, thank you very much. So next I will invite Doris from the WUSTL to present her research about what she's doing. Actually, I don't have any idea to introduce her to. So Doris.
Hello.
Hi.
Can everyone see? Can everyone see?
We
Hello?
see you,
Hello?
not the screen.
Oh, you. I'm sharing it. Okay. Sorry. I thought I shared the screen. Okay. Can you see it now?
No.
No. Oh dear. Can you see it?
Yes. we
Yeah.
can see it.
Okay. All right, cool. So, hello. Today I'm going to present to you a short summary of what I've been working on. And so I study locus olfaction, and what that means is that I study the neurological mechanisms behind the locus ability to smell, and that enable locus to encode chemosensory information in their little brains. In general, the ability to smell is important for all organisms, as this can tell them a lot about their environments. It can help them locate food or identify toxins and potential mates. And more specifically, I'm interested in how the locus olfactory system differentially encodes odors that also carry social information, such as pheromones. And so the amount and types of pheromones that a locus excretes is a function of what locus species it is, its age, and its behavioral state, like whether it's solitary or gregarious. And so whether it is in solitary, solitary gregarious state. And so, for example, there are pheromones like 4-vinyl anisole or benzaldehyde that have been shown to promote or inhibit aggregation. And chemicals like cis-3, non and 1-ol are excreted at levels that increase as male locus age, and female locus are more likely to mate with males that have higher levels. Furthermore, when locusts transition from solitary to gregarious states, there are phenotypic changes that happen to the olfactory system, but it's not well studied how this affects their ability to detect and encode pheromone sensory information. And so my work right now is to study how the locus olfactory system encodes this chemosensory information like pheromones. And so I do this using a variety of techniques, including electrophysiology, shown on the left, which involves opening the head of the locust and sticking electrodes into the brain while perfuming the locust with a bunch of smells such as pheromones. So the electrode basically records the electrical signal, like the neurons firing basically. And so in this way we can look at the in vivo natural activity of the neurons in the locust brain to study how the olfactory system encodes this type of sensory information. And I also use a grab bag or computational techniques to hopefully make sense of all this, these electrical signals that we record in the brain. And one technique that I'm highlighting here is the principal component analysis, as shown on the right. It's a technique that tries to maximize the variance captured in a data set. And this figure is showing the principal component analysis, or PCA, trajectories of 3 different odor presentations. And each odor is a different color. And so basically how the electrophysiological signals change over time for each of the odors. And a simple way to think about this PCA plot is that the closer 2 trajectories are to each other, the more similar the neural representations are, irrespective of the chemical structure. And so here, this is an example of one such data set. Each color represents the electrophysiological response to a different odor. Like each color is a different odor. The first 3 colors, so the 3 blues, are general odorants, and each of these colors, the neon green, yellow, and orange, are pheromone types. And so each, like I said, all the odorants have different chemical groups and properties, and so it makes sense that these all look distinct and they're like spread out. But if we look at it another way, so this is the exact same figure as the previous one, but in this one, I color coded it such that the general odors, so the non-pheromones are colored red and the pheromones are blue. And you can clearly see that the pheromones cluster together on like this half of the plot, and then the non-pheromones cluster together here. And implying that the electrophysiological activity in the locus olfactory system, that it has unique but still generalizable representations of pheromones and non-pheromone odors. And that is all I have to say. Hello?
Are you doing that in both? Are you doing that in both phases, or was it just?
No, I'm right now I'm using Americana species, so it's a non-truly aggregating. It doesn't have a gregarious state.
Okay.
Any other questions? Seems like Doris did a great job. No questions.
That's one interpretation.
So thank you, Chris and Doris. I think we don't have Chase to present the next one, so we have to go with the flow. So I will move forward with the next one. So Arani, can you please go ahead and explain what we are doing in the ECA symposium?
Yeah, let me just pull up my PowerPoint really quick. I have really bad internet service for some reason just in my office, but the
No,
rest
mine
of
too. I disconnected.
Okay, yeah, I ended up just logging back in, but let me share my screen really quick. So, not this one. Okay. Can you guys see that okay? Yeah. All right. So it's we're putting together a program for ESA. Right now it's myself, Alyssa, Odelia, and then please correct me if I pronounce this incorrectly. Is it Marine?
That is correct.
Okay. And Marine from ASU is going to be joining us on this. So this was a pretty last minute decision. The submission is due this coming Monday. So we're kind of scrambling this week to put it all together and finalize it as best as we can. So the ESA conference this year is going to be held in Vancouver, British Columbia, Canada from November 13th through the 16th. And the topic or the theme of this meeting is going to be entomology as inspiration, insects through art, science, and culture. So as the BPRI, and as we've seen through the last two presentations, we have a really broad range of topics that we discuss in terms of phenotypic plasticity. So for our program, what we're proposing is a phenotypic plasticity program from genomes to ecosystems. So bringing in speakers who can talk through a variety of all of these different things that interest all of us. So some of us are interested in genome and chromosome sequencing, others in nutritional, some are behavioral or developmental and such. And so we first came up with a list of potential speakers and then sent out invitation emails to those top 8. As of now, we have 5 confirmed speakers of the 8 that we've contacted, which includes Dr. Steve Simpson. And as we all know him, he's, locus person, does nutritional, physiological, and behavioral stuff. Our second speaker who's confirmed is Dr. Joel Kingsolver, who's a pretty big name in phenotypic plasticity. He has agreed to come to the conference. We just don't know if he's going to be in person or virtual. Then we have Dr. Karen Vanderberg. She's also going to be in person. She is actually in British Columbia already. She's at UBC. She's going to be talking about the genomic architecture of developmental plasticity in butterflies. And then we have Dr. Barani, who is also going to be in person, studying grasshoppers and doing sensory systems in these grasshoppers. And then our last confirmed speaker is Dr. Laura Levine at Washington State University, she works with dung beetles, specifically the development and evolution of adaptive phenotypic plasticity. So we have those 5 confirmed, and then 3 invited speakers that are unconfirmed. Dr. Ihab Abuhef in Canada, Drs. Anna Childers and Olga Duchenko, and then Dr. Julia Lesnick, who's an anthropologist who specifically focuses on insect consumption. So what we need from you, or ways that you can help, is if you know anyone who may be a good speaker to be included in this, contact us and let us know. And in the chat after I close my PowerPoint, I'm actually going to send a Google link so you can just submit the names through that. We are hoping and pushing for more women in STEM or minorities in STEM and early career scientists. So if you have anyone who fits those categories well, then definitely send them our way. And because we're doing, so many different varieties of phenotypic plasticity, the only real thing or requirement they have to meet is that they study phenotypic plasticity in some shape or form. And because we have 3 unconfirmed spots, we have room for potentially 3 more, but also this is not until November. So there are potential COVID cancellations, travel restrictions, and things like that. So it'd be good to have at least a nice list of backup potential speakers. We also need a title for the program. So if you have anything that might be fun or creative that you want to suggest, send your ideas our way. And then finally, because the theme is entomology as inspiration, insects through art, science, and culture, we're looking for ways we can integrate all of these 3 things into our program. So one of the ways that we thought about doing this is by displaying students, faculty, or staff, or anyone really that is involved with art as it relates to insects or phenotypic plasticity, as well as photographers. So if you are a photographer or an artist, if you like to draw insects or take pictures of insects, and you would like to have your work displayed at ESA, let us know. We would love to do that for you. Or if you have any other ideas in terms of like poets, potential performances, spoken word, or music, we would also love to hear those. So that's what we have.
Thank you, Arani.
Yeah, I will drop the link to submissions for potential speakers in the chat.
And also you can have more like 5 minutes to share feedbacks in here too.
Yeah, so if you have any ideas or if you want us to share any work of yours or anything like that, let us know. Coming up with the name is going to be difficult.
Yeah. Actually, I think like if you can find some songs or something that you can have a background play before starting this session or something related to grasshoppers, it might like look like you think a lot about every aspect of this session.
Yeah, and I mean, the good thing is it also doesn't have to be grasshoppers. So if you know someone who touches on any of those things with any type of insect or even just dabbles into phenotypic plasticity, then even that's great. I actually was looking into maybe reaching out to one of the Native American tribes in New Mexico. A lot of them have really strong backgrounds in like cicadas and how they're the original like origin of life and things like that. And so like traditional storytelling in that sense, something like that might be of interest.
I know Thailand is having like a lot of like insect eating culture. Like if someone know that kind of recipes or some photos or how that came to culture to eating something, that would be also cool. Because I think we are covered the scientific theme pretty much very nicely. Art and science is the one kind of we need to catch up a little bit. So maybe
can we have like an insect cooking class?
So we actually have
serious question.
Well, no, that's a great question. We've actually discussed this. So specifically, Hojun brought up that, we have a bunch of crickets and a bunch of locusts, and, he'd definitely be up to frying some with some salt and pepper and offering them as like, a little side snack at the event or something. So, and that's why we reached out to the anthropologist with that in mind, because she focuses a lot on like crickets and grasshoppers in diet. So yeah, you're not far from where we've actually been kind of dabbling a little bit.
But like, just like how we can invite an artist, can we invite a chef that specializes in cooking these things?
We could. That's actually not a bad idea. So a chef that specializes in insect cuisine. We also talked about maybe having like maybe a small snack session that includes fruits or veggies in the shape of insects, you know, like piling up a bunch of grapes and like adding little sticks on the side, making them look like caterpillars or things like that.
Like ants on a log.
Yeah, yeah. And the good thing with applying for the program is that if we don't get selected for the program, which is the highest tier that you can submit for the conference, you automatically get considered for the following one. And if you don't get that, then you're considered for a member one. So hopefully if we don't get the program, then at least we can end up in one of the other two smaller categories.
And also for the form, I think you have to be in your owner's organization. So I think only TAMU folks can open that. I don't know, not sure.
Only, what was that?
I cannot open the forms. It says that you need to have permission. The form can only be viewed by users in the owner's organization.
Oh,
So I think
let me
only
change
your,
that. Is that for the, is that for
Google
the
Forms.
form?
Yeah.
Okay. Let me change that really quick. Okay, let me try that again and see if it works this time.
Not on my side.
No?
Mm-mm.
Hmm. That is really weird.
Okay, while Arani get to that, just have a little bit, because we have another 10 minutes before we're closing, I would like to have some kind of convocation about the IDPP process, just to remind some and get some feedback from you guys. So okay. So first one is, I need all of you agreed IDP by 28. That's mean you have to have a meeting with your advisor and get your goals are agree with your supervisor that this is the one you will be accomplished by the end of October this year. Okay, please submit it by 28. If you're having trouble to get them have a meeting, let me know in advance, then I will make a note on there. And the next one is, I like to ask you to give the feedback through the Slido. So please scan this one and go to Slido. And first question is, this is just open. So let me know, how do you feel like doing this IDP process for the BPRI? Can you rate it from 1 to 5? I share the link from the two. Okay. The next one is, I know that some people would like and some people not like the old process. So what is your general feedback? You can share with the like verbally in here and what you need to change in this process. But if you have specific comment, you can put it in there too. But let me know what you overall think and where should we have to change in this process. Can I go with the names or any volunteer to share the feedback?
I can share my feedback
Mhm.
on the IDP. I mean, I know I did the example one, so I think it
Mhm.
was a little different, but personally for me, I thought like the very end, like saying strengths and weaknesses
Mhm.
was kind of like a, I don't know, it makes it more daunting and more, I feel like trying to say, okay, identify your boss's weakness and stuff like that when you go into the meeting and everything. I guess I would phrase it not as weakness, but maybe like something more subtle and everything like that. And it takes like anything personally out of it. But I thought it was overall like an okay thing. I feel like I didn't get, for the amount of time I put into it, I feel like I didn't get a great benefit. I think it benefits the PIs more than it does the students, but also depends on some PIs, I think probably put in more work than others. So I think it was good, just not a per out like time put in versus output kind of thing.
Okay. So anyone else have any feedback?
I will say IDPs are helpful. I mean, we do it here at ASU as well. Whereas I think IDPs are helpful. I think it helps you understand what you want to accomplish a little early on. So you kind of gets you thinking early on instead of very late. So that's helpful. And if you and your PI can stay on the same page. But I think also at the same time, doing it early in your career can be also daunting because it does ask some questions about career and stuff. And you're like, oh, I'm right now trying to figure out my dissertation. I don't know about my career yet. So I think that can be a daunting aspect of an IDP, but I still think that really gets you thinking about it. And it is very helpful to keep you and your PI on the same page if you don't see them often.
Okay. Anyone else? Can I, if I call upon the name, is it be okay? Or I don't like to put it into the place. So
I have to agree with Chris on my experience with that too, that I think I put in, I think, well, going the other way, I think it was more beneficial to me than it was to discuss it with my PIs. It gave me a better outline of what I was going to be doing, but I didn't have enough time to actually discuss that with my PIs because our meetings are only 30 minutes. And so we have 30 minutes to get through an entire IDP. So my comment for things we could change is to maybe focus on working and discussing sections of the IDP at a time, as opposed to trying to get through an entire IDP in one sitting with your PIs.
okay. Anyone else? I would like to see from other university, not TAMU, Doris, or Alexis, or Jai. Can someone share some feedback?
Do you want feedback on the IDP specifically, or
Yeah,
just
so
like,
what do you feel about that, doing it, whether it's useful or what, do you want to change something?
I thought
General feedback, it's
useful.
not need to be specific.
I thought it was useful. Because I'm not really good with writing down my goals and stuff and putting timelines. So I think it was good for just having general structure for myself. But since I'm only like a 2-year master's student, some of that stuff wasn't applicable for me. And I do see my PI weekly, daily, really. I stop by her office every day. So we are in tight communication. And so some of it kind of felt redundant because we talk about it frequently.
Okay, so in my sense, what I learned that from, because I review the IDP, not only yours, because earlier when I'm coming to this job, I evaluate the IDP from some student in TAMU context. So they says that it's really benefit if they started earlier, because for them it was introduced like 3rd or 4th year, then course selection, everything they have completely done. So they didn't have the career choice, so they was feeling like lost. So they say it's earlier stages benefit, but we are still learning by doing the process. So definitely we'll see how it goes from the 2nd year and 3rd year. And one thing I think need to emphasize as trainee, I think having you guys at least have some communication with your professors that you are interested in this career will help you in near future in some way. That is what I generally get from the whole process, because before IDP, I didn't hear anything about your careers from your PIs. Now when they're talking about something implementing to you guys, say like, maybe we need to think about their career choice, because we have some people don't like academics. So I think there is some like 50-50 kind of advantages and disadvantages, but I really like your honest feedback on where we need to change. Hopefully from the next evaluation, next year IDP meeting, maybe a little bit structured one, then everyone have the equal chance, because some people have 30 minutes communication about, and some people get very long length of very detailed meeting with the PI. So I think we need to have kind of equality accessible to everybody, regardless of mentor's schedule is busy or not. I will get that in my mind in next time. So the final thing that I wanted to share is for the next meeting. So you don't need to mark your calendar now, but if you can, please mark your calendar in here. And also, about today meeting, please rate it, because I'm recording each meeting, how it's improved or not improved throughout the time. Thank you. So I know that this time we able to get a little bit of the attendance from most of the people that we didn't able to get it in the last meeting. So thank you very much for spending time and joining the meeting. And I will keep this recording posted who have missed the session today. And hopefully next meeting we will have more people joining and was able to present, because this time I was really hard to find people to present. So thank you very much for your time, and see you in next month. Bye-bye.
Bye.